record.
Okay. So it's 8:01, so maybe we'll go ahead and get started. We have the pleasure of having Dr. Jill Blumenthal today. I'm going to read her bio, but improvise just for fun. So Jill Blumenthal is an associate professor in the Division of Infectious Diseases and Global Public Health in the Department of Medicine at UC San Diego Health. She conducts clinical research on HIV prevention using PrEP in cisgender men who have sex with men, cisgender women, and transgender and non-binary individuals at risk for HIV. She sees adolescents and adults living with HIV and is also the director of PrEP services at the Owen Clinic. Jill also cares for transgender non-binary individuals seeking gender-affirming care at the Owen Clinic and recently established a gender health program at UC San Diego Health. In 2018, she co-founded the Transgender Community Action Advisory Board to engage and empower the transgender non-binary community in research. Jill currently serves as the co-chair of the UC San Diego Chancellor's Advisory Committee on Gender Identity and Sexual Orientation Issues, and was recently selected to be a UCSD LEAD, which stands for Leaders for Equity Advancement and Diversity Fellow, to advance UCSD's vision of cultivating a diverse and inclusive university community that encourages respectful, open dialogue. Jill has been a faculty with us for many years now, and I think is one of our certainly most energetic people. And I think the word that describes her I chose is pizzazz. I think pizzazz is, energy, but, I think it's a very positive kind of vibe. And so she's very well liked by her colleagues, us. I am not paid to do that, as well as her patients. I think she really embodies what we like to have in terms of our faculty members at the Owen Clinic. And it's been an absolute pleasure to have her work with us. And she's definitely the go-to person when it comes to issues surrounding PrEP, as well as the care of our LGBTQIA+ individuals. So I'm glad to introduce Dr. Jill Blumenthal. She's going to be talking about an update on CROI 2022, more specifically on HIV testing and prevention. Go ahead, Jill.
Dan, that brought a tear to my eye. For real. That was really sweet. Thank you. I was talking to Gabe Wagner last night, and he made a comment about my energy. I'm like, you should see me when I get home, though, because it's just like plop. So I guess I use it all up during the day, and then it's done. But I appreciate that. Yeah, I hope that I can do some coverage for everyone who both were and were not able to attend CROI. This definitely favors HIV prevention. There's a little bit on testing that I wanted to talk about. If you are willing to turn on your camera, I like looking at faces. I keep some up at the bottom. So whoever wants to, it's nice for me to know that maybe someone's listening, but no pressure to do so. So with that, I will get started. I do have Gilead funding to the institution, was on a VIV advisory board last year.
And I'll monitor your chat bot, and if an appropriate question comes up, I may interrupt you.
You mean not Gabe's haha? Because that was important. Just kidding. No. Okay. So here's what I'm going to go through today. HIV incidence in the US, we have some new updates from the CDC. This just says HIV testing, but it was an interesting HIV testing study that was done. I think probably what everyone continues to be eager to hear about is long-acting cabotegravir for PrEP, and there were a few more studies on that. Islatravir for PrEP, a few studies there. There were some vaginal ring studies. There was definitely a good amount on PrEP and transgender individuals, so I was going to go into that. And then just very briefly about the HIV-1 vaccine that was presented. So the CDC presented the estimated lifetime risk of HIV diagnosis, and this was data between 2017 and 2019 from the National HIV Surveillance System. They also, though, used the National Center for Health Statistics and the US Census to calculate the probability of diagnosis of HIV. This is very binary. And that is, they were really dividing it. This is the data that they have. But this is broken down into male sex and female sex. So overall, people in the United States run a 1 in 120 lifetime risk of HIV. Now, what I want to highlight are the subgroup risks, which I don't think will surprise anyone, but this is not broken down by behavioral risk, which is just a different way to look at it. So Black males have the highest risk at 1 in 27, followed by Hispanic males at 1 in 50, and then Black females 1 in 75. And again, data on gender identity is not available. So, I don't know how much that would change things. These are obviously just big, broad buckets and does not consider exposure routes, risk, or networks. And then what they also did was look at the lifetime risk of an HIV diagnosis by state. Obviously, we know that people move in and out of states. So, what does this mean? But obviously just, I think hammers home again, that we're seeing a lot of the highest risks. So the darkest colors indicate the highest incidence or the highest lifetime risk. So those are mostly the southern states, New York. Oh gosh, is that Utah or Nevada? That's embarrassing for me. We'll call it Nevada, and I hope that's correct. Okay, good, I got a nod. And then, California is actually really, what's been interesting about California, I think over the last few years, is that it really has sort of, it's the average in a lot of ways, and that's exactly where it is right now. So this was an interesting HIV testing study that was done. It was really, it was a CDC program, direct to consumer distribution of 100,000 HIV self-tests. And they designed this based on earlier evidence that internet-based approaches to connect with people and mail them HIV tests was really high. So this was what they wanted to do. So the goal was to deliver 100,000, and they were going to do it over about a year, but that the demand was so high that they did it in 9 months. And they went to all 50 states, Washington, D.C., and Puerto Rico. 55% of the orders came from EHE priority locations. And I think this is what is if you're going to remember something, this is the interesting thing to remember. So 1/3 of test orders had not been tested in the past year, and 1/4 had never been tested. So this did reach people who perhaps, hadn't tested, although there was, they did go through it, and there were a lot of people who requested tests who, the risk was probably extremely low. It's hard to know. That wasn't all accounted for here. But so cisgender MSM placed a little less than half of tests, and just about 20% of those people never had an HIV test. And cisgender Black women accounted for 11% of all orders, of which, again, about 20% never had an earlier test. Transgender women, very small amount at 1.6%. And you could, so you were encouraged to take a short survey, and you could reorder in 90 days. So a lot of people ended up being repeat testers. So that's why you only see 57,000 residents who were sorry, of the 57,000, it was because some people requested second tests. There's no information on what the test results were. So it was just a program to get it out to people. And I'm not sure if they will continue to do this, but obviously had very high uptake if they were able to do it so quickly. Here is, I hope this, I mean, this is a very overwhelming slide. This is, this is by AVAC. This is really just to look at what is coming for HIV prevention, some of which I'll talk about today. So where we are at right now, or at least in phase 3, 3B, and 4, we have the monthly, so it took me a while to go through this, and you'll be able to see my slides so you can go through this more, because you have to match the color, which is a little tricky, especially if your color discernment isn't fabulous. So this is the dapivirine ring. This is cabotegravir. This is Islatravir. This is Longacting, and this is TAF-FTC. And I do want to note, and I'll talk about it in a moment, I don't believe this is correct, but both the Merck and Gilead drugs are currently in trials that are paused, and I'll talk about that a little bit later. Figure also doesn't include BNAbs and vaccines in development. I think this is just exciting. There's a lot coming. There's a lot of choice. I think we've talked about this before. Choice seems to be the most important thing when it comes to prevention, because, or one of the most important things, as if people don't see themselves to be at risk, or it's not on the forefront of their mind, being able to do it in the easiest way that works best for them is probably what will work best overall. So do want to just go through quickly what has been approved. So I will skip, everyone knows about PrEP, everyone knows about oral tenofovir based PrEP. I'm talking about PrEP. And the formulation, dosing, the options are daily, now 2-on-1, sort of permissively in the new FDA guidelines, or the new CDC guidelines. And just so people know, because this does come up, there is a study of Tanafavir, TAF-FTC for cisgender women in Purpose 1. There are a few sites in the United States, but none on the West Coast. So that should be nice, so that we can actually offer cisgender women the same things that we offer our other patients. We know about PEP, long-acting cabotegravir, which was FDA approved in 2021. And with some analysis, and for those who saw the New England Journal of Medicine article, it was more than 66% efficacious compared to FTDC. But then they've also done analyses where they look at counterfactual rates, and that's even higher. So we know that long-acting injectable cabotegravir works very well, and we are starting to roll that out, certainly at UCSD and in other places as well. The dapivirine vaginal ring, which is a monthly self-inserted ring, works efficacy of about 50%, which was why it was drawn from FDA review in November of this past year, maybe with not a lot of noise, but it is recommended in the WHO guidelines. I won't be talking about this one, but one of the very exciting ARV-based prevention options are the dual prevention pills, which combine both protection against HIV and pregnancy. This one is a daily oral pill with levodopa. I'm so bad at this. This is great. We'll just call it Ling, but know that it's a long word for contraception, plus ethinyl estradiol. And the HIV incidence reduction was greater than 90%, maybe available in 2024. I think that is extraordinarily exciting. Sorry to the OBGYNs if I butchered that. And then lenacapavir, well, we have two more. Lenacapavir, which is a Q6 monthly injection. There are two studies, Purpose 1, Purpose 2. Purpose 1 is in cisgender women, young cisgender women in South Africa, and then Purpose 2 is with MSM, trans women, trans men, non-binary individuals, of which we are a site. And then Islatravir, which is a monthly oral pill, and it has the potential for a yearly implant. The two studies that were going on were Empower 022 and Empower 024, and different sites in different places. Obviously, we don't know what the HIV incidence reduction is because the studies were ongoing and now clinically on hold. And I'm sure a lot of people saw this, but just in case you didn't, so there was good and bad news in December. The good news was that cabotegravir for prevention got approved, which was really exciting, pretty game changing in this field. But two of the big studies looking at different agents were put on hold. For lenacapavir, it was due to concerns about the vial compatibility. And Gilead is working as quickly as they can to try to resolve that issue to get the trial going again. And then Islatravir, there was a clinical hold due to concerns about decreases in the lymphocytes and CD4 cells. Obviously of great concern for treatment trials, maybe less so for prevention trials. I don't know for sure, and I'm paid by no one, but I think this will, I hope that these will both continue as they are great options, just different ways to take medications for people. And this was from Chloe Orkin's plenary, and it was fabulous. So if you're able to go in and watch it, I would definitely recommend that. Okay, so HPTN 083, they did an updated efficacy and safety analysis. Just to remind people, this was a randomized double-blind phase 2b/3 study. 083 was the one that included MSM and transgender women. The first step was an oral lead-in, and then at week 5, they were switched to either long-acting cabotegravir every 2 months and placebo, FTC TDF, or the other way around. And then this was the blinded area, but then people became unblinded. The unblinded period, or they got unblinded at about 1.5 years of median per participant follow-up. And then the step 3 was just the oral medication for a year. And just to refresh everyone's memory, they previously reported 13 infections in the CAB arm versus 39 in the FTC TDF arm. One incident CAB infection was re-adjudicated to a baseline infection. So actually it improved the hazard ratio to 0.32. And so this analysis looked at additional infections after 1 year of follow-up after unblinding. So again, this is so there were 13 additional infections identified on CAB, but 2 were in the blinded period. So they're not shown here in this unblinded period. And that was part of what I just talked about, what they found. And then there were 11 during the unblinded period, 1 with on-time injections, 3 with delayed injections, and 7 with greater than 6 months after cabotegravir. So that brings them to the overall efficacy of 25 infections in the CAB arm versus 72 infections in the CAB arm, or in the, TDF FTC arm, which still shows a statistically significant difference favoring CAB. So just, the higher incidence in the 1-year unblinded analysis compared to the primary analysis, there was some thought that maybe this resulted from decreased TDF FTC adherence, right? Because you had more infections in the TDF FTC arm, as well as reduced CAB injection coverage. And then there may have been higher relative contributions to overall person time from high incidence regions that were later enrollers. So that was their thought there. So, and just this is, I think, is a big number. There were a total of 7 breakthrough infections on CAB despite on-time dosing. So that's relatively rare. That's an incidence of 0.15 per 100 person years. They've done a very careful case-by-case analysis, and they still have not been able to discern a reason for these breakthroughs. But with all of this taken together, and I had already said this number, with the 25 incident infections over a little shy of 5,000 person years, there's a 66% reduction in incidence for CAB, LA versus TDF FTC. So this was very big and exciting news. But so what had come out before and what was, concerning to people were the infections that occurred with, the participants that had received it and maybe received the CAB, but the antigen antibody test didn't detect the infection. So there was treatment initiation delay, and there were emergence of INSTI-RAMs. And of course, this is of great concern because it could compromise our most basic and fundamental ART regimens. So this was a study using a single genome sequencing INSTI assay in HPTN083, where they had 7 participants who had received it. 5 had INSTI resistance and 2 weren't able to be genotyped because their HIV RNA was less than 500 at all visits. So they used the single genome sequencing to see if they could determine whether earlier infection of HIV would occur, and then prior to the appearance of the RAMs. So they had 21 samples from these 7 people. They used the Stanford HIV resistance database. In 6 out of the 7 patients, major INSTI-RAMs were first detected in samples with HIV RNA less than 500. So this, this is why our guidelines have changed, particularly for cabotegravir, that screening with an HIV RNA assay would have detected infection before a major RAM occurred or accumulation of additional ones, which happened in 2 of the cases. So their major conclusion is that the use of a sensitive RNA assay for HIV screening for long-acting CAB could improve earlier detection of infection, earlier ART initiation, and then reduce the risk of developing INSTI resistance. So, and then there was a comment about this because, the guidelines have come out, the new CDC guidelines have now recommended that you do an HIV RNA for all people taking PrEP, regardless of the formulation. And I think it's felt by a lot of people in the PrEP world that is an overcall and maybe a use of resources that shouldn't be. This data suggests that you definitely need to do it for people taking long-acting cabotegravir. I don't think the data is there for oral formulations. But then the other point is who can actually get RNAs, and that's not easy in a lot of places. They concluded that because it's so highly efficacious and better than what's available in a lot of countries, a lot of middle and low income countries, that long-acting cabotegravir PrEP should still be considered even when you can't get HIV RNA screening, really, because of the difference compared to TDF FTC.
Jill, there was a comment from Shannon for the 2 patients with less than 500 copies. Had they been started on antiretroviral therapy, or was that still just on cabotegravir alone?
It was just cab alone because their antigen antibodies. Oh, you know what? I'm not sure. It was definitely delayed though. I know that they couldn't detect the RAMs, but no, I think these were people. I think they were continued per protocol, which would have been to continue on cabotegravir. So this is why these people developed them. I hope that answers the question. I believe they just stayed on cab alone. Thanks. Okay, just important to always talk about pregnancy. So HPT and 084, women were required, so the cisgender women in that study were required to be on long-acting contraception. And remember, this study came about during the time where there were huge concerns about dolutegravir and neural defects. So this was a really important thing to be looking at. So after pregnancy was diagnosed, participants were unblinded and no further CAB was received, but there were women who received CAB-LA until the pregnancy was diagnosis. And the big key here is that there were no congenital anomalies. However, and take what you want from this, there were more AEs in the CAB arm compared to TDF FTC, but they were different AEs sort of all over the place. But certainly if you see an N of N equals 9 and an N equals 1, you might think something, but they were all different sorts of AEs. And then just quickly about the CAB concentrations, they were similar for non-pregnant women and pregnant women, but a higher BMI was associated with a longer half-life of CAB. And HPTN 084, the open label extension, does not require long-acting contraception, which definitely is a barrier for a lot of individuals in studies. And I think this data showing, no congenital anomalies was really helpful for that. So the long-acting injectables for prevention in HIV and unplanned pregnancy is really this new frontier that, I mean, not so new, but still, we're still in phase 1 and phase 2s of phase 1s, phase 2s of development. And this group, primarily out of UNC, proposed to develop an injectable, long-acting, biodegradable, and removable in situ forming implant. That is what an ISFI is, and you'll see that on this, as a multi-purpose prevention technology, MPT. So in phase 1, they looked at dolutegravir plus a contraceptive, which showed feasibility and ultra long-acting release. It could be removed. And there were PK and efficacy studies in humanized mouse models showing protection against HIV. In the next phase of this one, they wanted to look at it with long-acting cabotegravir. So that was what was looked at here. I am not a pharmacist or a pharmacokineticist, but I'll basically just tell you what's important to take away here is that the ARVs were above their 4 times the IC90 threshold for 90 days. There were no significant differences in the ARV release when formulated with hormones. Dolutegravir, CAB, and MPA elicit 0-order release, whereas ENG elicits 1st-order release. And that CAB ISFIs have shown that they can actually release that 4 times IC90 for up to 6 months in macaques. So the potential for doing this even longer. What is not shown here, but other slides that the presenter had, was that local inflammation was assessed from staining, and there was just mild to non-existent inflammation after 30 to 90 days. And they also looked at TNF-alpha and IL-6 through 90 days, which showed, again, mild systemic inflammation. And future studies for this will be to be looking at the PK tail, and then also the next step would be PK and efficacy in non-human primates, which is obviously a bigger step since we're talking about mouse models here. So let's move on to Islatravir. So this, as some people will know, Ryan Racine, if he's on, this to me is such an exciting drug. The idea of being able to take a monthly oral pill seems fabulous for a lot of people, maybe in particular for cisgender women who don't view themselves to be at risk. This was a study of seronegative individuals who are at low risk for acquiring it. And this was to look at safety and tolerability. I hope that's what this is, sorry. Yes, this was the metabolic and renal outcomes, which are obviously really important, as that's been more of an issue for Tanafavir FTC. And you can see they got actually a good number of individuals assigned female at birth with a pretty good range of race and ethnicity. Basically, there were 3 groups of Islatravir 60, Islatravir 120, and then placebo. And basically what they found here at week 24 was that there were no clinically meaningful differences compared to placebo in the metabolic and renal parameters with either 60 or 120 milligrams after 6 months of doses. But again, when this was presented, they said that the program was placed on hold because of the lymphocytes. So this is where we are, and things are stagnant right now. Another presentation by Craig Hendricks on Islatravir, same study, from the same study, but a subset. They looked at blood and mucosal distribution. Again, a very highly dominated cisgender women study. It's a phase 2A trial for PrEP. And basically what they found, great news, plasma Islatravir is highly correlated with Islatravir triphosphate in cervical, vaginal, and rectal tissues, which is great news because you'd have a great surrogate. You could use plasma PK measurements as a surrogate for some tissue exposures. Again, on hold now. I'll pause for a second. I didn't see any more questions come up, and I'll keep moving. So no, I was going to pause. I can't say I'm going to pause and then keep moving. It's contradictory.
No questions.
No questions. I'm going to sip my coffee for a second. Okay. So the next study was by my dear friend and colleague who spoke at our high rounds last year. He talked about a phase 1 PK safety and acceptability study of a 90-day Tanafavir vaginal ring. So a new formulation, although Tanafavir is not, not new to us. But the reason, that they thought about doing this, that there were, several studies that looked at Tanafavir topical formulations, some of which had some signals that maybe this could be useful. And, an extended duration ring could help increase adherence, maybe effectiveness, and then also reduce cost and clinic and user burden. So this was a 2-arm randomized 2-to-1 trial that used a 1.4 gram Tanafavir polyurethane ring versus a placebo ring. There were 49 individuals who were healthy, and there were 3 study sites. This is just showing you what the rings look like at different times, depending on how much medication is still in there. So in terms of the safety data, there were no statistically significant differences in the proportion of participants with grade 2 or higher GU adverse events in the Tanafavir arm compared with the placebo. And then there were no grade 3 or higher AEs. There were a lot of AEs, and they were, but they were mostly unrelated to the unrelated to the ring. So the ones that were related, and make of this what you will, they were, vaginal discharge, there was candidiasis, odor, uterine spasm, dysmenorrhea. I don't know, when I saw this, I was like, oh, that's a lot of stuff. So I think, that's something that they will hopefully continue to work on as they develop this. But the idea of having something that has longer formulation, obviously quite appealing. So then looking at the PK data for this study, this actually was really important. They found that the Tmax was 34 days in cervical vaginal fluid and rectal fluid, but longer in plasma, and the mean Tanafavir concentrations declined at day 91 across all compartments. So actually, so they declined it at day 91. Acceptability, people really liked the ring. There were a few people that, that that didn't like it, but obviously clusters more to the higher end of liking. But again, what I had mentioned was that the target cervical tissues were exceeded, but the tenofovir concentrations declined by day 91 in a subset. So they'll probably look at some higher release rings to determine the optimal duration of use. So 3 months might be a little bit too long, or they need to change that formulation. But this is a start of looking at rings that can stay in for a longer period of time. This was a study done by Kenneth and Guri in young African women in the REACH study. The REACH study enrolled just about 250 HIV negative non-pregnant adolescent girls and young women between the ages of 16 to 21 from South Africa, Zimbabwe, and Uganda between 2019 and 2021. They were randomized to either the monthly ring or the daily pill, and then they were crossed over for the second 6 month period. So first 6 months, second 6 months, and then there was a choice period, and they got to choose either the ring, PrEP, or neither in the third 6 month period. So what they found in terms of product choice in that third period, when they said whatever you would like, so of the 92% who reached the choice period, 2/3 chose the ring, a third chose oral PrEP, and then 2% preferred no product. There was so high adherence, and this is the oral PrEP adherence. So where there was high adherence, and that's where there was always green tenofovir diphosphate levels of greater than 700 femtomoles per dBs punch, that was highly associated with the choice of oral PrEP. So if you were, I don't know which direction that goes, but obviously if you don't have much of a problem taking it, then you are likely to choose it. I also want to note that they determined high adherence in cisgender women to be greater than 700 femtomoles per punch. I think this is good news. It seems like there's been some consensus among people that the greater than 700, which was really just used in cisgender men and actually just cisgender men, I will say, now may more comfortably be able to be used in different populations, including cisgender women. I still haven't seen, and correct me if I'm wrong, I still haven't seen a study outwardly saying that, but it does seem like the threshold in a lot of these studies are now saying that the 700 mark, which is the equivalent of about 4 doses per week, would be acceptable and considered highly adherent. And again, in terms of ring adherence, they didn't find that same association, that if you were taking the ring well, that you necessarily chose to continue the ring. But I think what's also really important, you can see these always green numbers are relatively high, and they indicate partial to high adherence in both the ring and the oral PrEP, and much higher than prior studies. And the reason they focus on adolescent girls and young women is these are the groups that have struggled the most. And what they concluded was that adolescent girls and young women can make informed choices about prevention products and are motivated to continue the use of a product of their preference. So just to say let's not make, this group can do this. So they're just ways that we have to reach them and make sure that there is choice for them. Oh, this was just some abstract. No, I was going to make a funny joke about this is the one and only one you had to see. This was my first oral abstract. I will talk about this at a later one, so I'll just give you sort of the high level overview of it. So we looked at the bidirectional effects of hormone therapy and PrEP in transgender individuals. This has been studied before, but most of the studies are very small. The time period follow-up is usually just about a month, and hardly any transgender men have been enrolled in these studies. So we took from Sheldon Morris and David Moore's IM PrEP study, where we had 256 individuals randomized, and then we enrolled some of those individuals into a sub-study. And I think you can see my arrow, that this is sort of the most important point. We ended up getting to 39 transgender men and 49 transgender women on stable hormone therapy that we could look at. Because the biggest question in the scientific world is, does this affect your PrEP? I'd say the bigger issue for people who are taking these medications are, does this affect my hormones? We also looked, we did something a little bit different, where we looked at desired hormone effects and satisfaction with your hormone therapy at week 0 and 24. I said I was going to do this quickly, and I'm going through the whole thing. The highlight is that in the 49 transgender women on stable estrogen, so this is the one looking at the estradiol concentration. So those who are taking stable estrogen, the estrogen levels did not change in those who are taking PrEP. So no effect on the estradiol. In the just shy of 40 transgender men on stable testosterone, the concentrations were lower between 0 and 12, but they weren't statistically significant. And it was encouraged that we measure Cohen's D to look at the effect size of hormone concentrations, and the effect size was very small. So this was good news, although a little bit different than what had been seen in other studies, in terms of maybe some numerical change in the testosterone concentration. This is the part that I think the scientific world gets really excited about. Looking at week 12, were you on hormone therapy compared to, were you not on hormone therapy compared to being on hormone therapy? No difference in the tenofovir diphosphate level. Similarly for transgender men. So transgender women here compared to transgender men, no difference. And actually for those in the field, these are pretty robust numbers too. They're very wide confidence intervals, or large standard deviation, but these are really big numbers. So, and then I think most importantly, and how I can reassure patients, is that there was no change in their body image satisfaction and their satisfaction with hormone therapy when they were taking or not taking, when they weren't taking PrEP at baseline to when they were taking it at week 24. So what came, there were a bunch of questions for this that I think was very exciting for us and what we'd like to explore. I just talked about 4 doses potentially being an option now for, or it seems like the world has settled on that for cisgender women. And then everyone asks, well, what about transgender individuals? Do we keep telling them they have to take it, 7 days a week? And our data would, and we need to look at this, and we will, but with these numbers being as high as they are, it seems like there probably is some room for forgiveness, with the estrogen and testosterone really not having much of an effect on people. Now, that being said, this was not a PK study. The measures were drawn, or the tenofovir levels and the estradiol and testosterone levels were drawn at random. And we had a lot of dropout in the study, but we still had a lot of people in it. So that was that. There was another study looking at HIV testing and PrEP among transgender women and men. It was it was a strange analysis that I think had some encouraging findings, but I'm not sure how it melded all together. But they basically looked at the market scan database from 2012 to 2019, which is weighted for nationally representative information on diagnoses, procedures, and prescriptions for health services. This was by Karen Hoover's group, who does a lot of really interesting work with prescriptions and looking at, diagnoses and all those sorts of things. And what's great is that the weighted numbers of people who had transgender related codes for, gender dysphoria or other things, increased from about 8,000 to 94,000 in 2019. So obviously there are more people that are being seen for these things. It's being recognized. And then similarly, the weighted number of transgender people who were prescribed hormone therapy also increased in that time, from a little under 2,000 to 38,000. So we're seeing, lots more gender affirming care happening. And then what you're seeing on the right-hand side here is the the weighted numbers for testing, HIV testing. Yeah. No, these are the unweighted testing. Sorry. So unweighted testing. And what you can see is it steadily increases over time. In 2012, we were hardly doing this and hardly recognizing it. And it's really gone up a lot. And then the less clear bars are PrEP, are who actually were taking PrEP. So we're starting to see some increases there. But their conclusion, and this is what I struggled with a little bit, was that HIV testing, I understand what they're trying to get at. They were saying that HIV testing and PrEP prescriptions for patients who use gender-affirming hormone therapy increased over the study period, but there were many prevention opportunities that were likely missed. I think it's hard they didn't overlay this data, so I think that's kind of a big statement to make. But I think the point is we're definitely getting more people seen, who are carrying these codes and are being prescribed hormone therapy. Yet our testing is going up, but our PrEP offerings are still low. So maybe we're missing opportunities. Take what you will from that. I actually asked a question about this and it was never answered. So, but I just thought it was cool to see at least all the progress that we're making for transgender individuals in terms of being able to access care better. And then this was PrEP need and services for transgender individuals in the Thrive project. So Thrive supported 7 health departments to lead collaboratives of CBOs and clinics for HIV prevention and care. And this to me seems like a real Herculean effort. They enrolled close to 1,000 transgender women who were Black and Hispanic, and just about 400 Black and Hispanic transgender men. I'm saying Hispanic, that was how the authors wrote it, or that's how they grouped individuals. So they were really able to successfully engage PrEP eligible individuals. And then here's what this graphic shows on the right. So many were referred to providers, but like a lot of what we're seeing, the drop off occurs when we're trying to link people to provider and then actually prescribe PrEP. And then of course, the argument that I make in our clinic all the time is really that most important part is who actually stays retained. You can prescribe someone PrEP, doesn't matter if they just take it for a month and then they're not on it again. But this was great, just trying to, get people aware of this. And, I've seen some data out there that getting people aware of PrEP and them thinking about it may ultimately be useful in getting them to PrEP even at later times. So I think this was wonderful that they were able to do this huge effort in this project. And then I'm going to close. I'm not a vaccine person, but I think this was important for people to see in Bakoto, which was a vaccine regimen in African women. It was a heterologous HIV-1 vaccine, which had the adenovirus 26 vector and it expressed mosaic, gag, pollen envelope antigens. For those who know a lot about these things. And it was, huge, big study, 1,300 people in the vaccine and the placebo arm. They did a month 24 analysis with possible continuation through month 36. And as with most of these studies, the primary endpoint was vaccine efficacy from months 7 to 24. And they also looked at safety and tolerability. And the big news from this is that the vaccine did not provide statistically significant protection against HIV. So it failed to meet the pre-specified criteria for continuing through month 36. And they looked at it in lots of different ways, I think, to see if they could find any kind of signal, and there was no signal to be had. So this was, the HIV incidence was quite high in this study, and the vaccine regimen did not provide the protection they wanted it to. They did say that there, for those, again, who think about this more, there is an ongoing phase 3 trial, Mosaico, looking at the efficacy of a regimen that's similar, but it has a modified boost of adenovirus 26 bivalent GP140 in MSM and transgender individuals in the Americas and the the Americas and Europe. The Americas includes the United States. So this is obviously still one of the big areas. I'm sure, I know a lot of us get questions, when's that vaccine coming? When's that vaccine coming? And unfortunately, this was another failure. But we'll keep the hope alive. And I just wanted to show you how I prepared for CROI. I first took out my my bearded dragon, Murphy, who helped me with my slides. And then this is me looking, I think it would be a C word, maybe. I looked very scared during the taping. And then that is my dog Tallulah, who watched me watching myself on video and was very confused hearing my sound coming out of a computer while watching me. So anyway, I just wanted to show you my little transition there. And I really want to thank particularly Al Liu and Catherine Koss. We shared some slides, but Clinical Care Options, I took a few from, and NATAP does a wonderful job consolidating this. And that is how, I'd recommend people going and looking if they want. NATAP has everything listed in all the slides with some great summaries of some of the presentations.
Great. Thank you, Jill, and
Sure.
perfect timing. We've got 8 minutes for questions. The chat pod was relatively quiet, so
I saw.
yes. So Darcy says amazing presentation. Thank you. Anyone want to unmute themselves and ask a question?
I'd say, or share something that I might have missed. This obviously was me trying to be as comprehensive as possible, but not overwhelming you. There were definitely a lot of other great presentations there.
Well, while people are thinking of a question, I was going to ask if whether or not you feel like studies are moving forward with asking more about gender identity and non-binary individuals. Is that something do you think that's a moving trend in that direction for people to start reporting data that way?
Okay, so I can, there are big parts of that and small parts of that, because I think there are some, there are mandates happening. So I think the next time that the CDC reports their data, I think they will have gender identity. This was obviously very, very binary. I think most places now are really doing their best to collect the data. Throughout CROI, it was really nice to see people actually getting the difference between sex, and sex assigned at birth and gender identity. So I think it's happening more. I think this is also an audience that's aware of this. So we're trending in that direction. I do believe that the CDC is now going to more formally require it. What I haven't done in a while, and maybe someone else can tell me, at least when you had to give the NIH your planned enrollments, the only options were to write female and male. And that to me was one of the biggest issues. I haven't done one in a while. I don't know if anyone can share if the NIH tables are still male and female. Oh, it's a quiet group.
Well, while we're mulling that,
those are all places, if they're requiring it, then you have to do it, and we should be doing it. Obviously, you know how I feel.
Yeah. And I think if you look in the chat, I think Nettie said, was there some misleading media on the vaccine? Multiple patients asking me about recently. Sadly, the reporting giving them false hope.
So I think maybe the reporting that people were talking about was the messenger RNA vaccine that was getting started, and then people were really excited about that. Obviously, that's still at the very beginning. I don't know if there was, I didn't see anything else on media. The Bakoto trial, this was the first time. I think I'd seen one thing about it, but I don't know. So I'm just wondering if they were maybe confusing it with the messenger RNA vaccine. I think Darcy asked a great question, because this was a very hot topic. The viral load monitoring prep, which can be very expensive, time consuming, and who knows what it means if you find a viral load that's less than 20, as we know and we've seen in trials. So to Darcy's point, I think it will stay absolutely for cabotegravir. I think the data has shown very good reason that the cabotegravir seems to suppress the antigen antibody response. So it does take some time to show up. I do not think it's appropriate in oral PrEP. We have not, at least, oral PrEP with tenofovir. It may be necessary for other regimens that come around, but we've sort of decided as a clinic, and people can do what they want. And I've discussed this with other people who do PrEP a lot, including the author of 083, who said, he thinks that the CDC guidelines were taken from 083 and applied to everything, which really shouldn't have been the case, because we know that there aren't PrEP breakthroughs the same way, and we're able to pick up infections relatively easily. So I think you continue to use the viral load when someone is, acknowledging they're poorly adherent, perhaps in someone with a lot of sex partners, and you have extra concern. Because they're in the CDC guidelines, they should ultimately be covered. For me right now, the greatest fear is that I do a viral load and someone gets charged for it. So I'm being very cautious about how I approach it and really doing it on a case-by-case basis.
Great. And I think Shannon has a question. Any changes to how you approach PrEP prescribing based on this? So a broad clinical kind of question.
No, I think Shannon, I think you hit the nail on the head. I alluded to whether or not, we can, because I think a lot of people know that, we do 2-on-1. And when I say we do it, we tell people it's an option. And we offer, or we say that 2-on-1 also could be appropriate using Descovy or, tenofovir alafenamide based regimen, even though it hasn't been studied in that. For MSM, for cisgender men having sex with men, or bisexual, gay and bisexual men, essentially. Some of the data that came out makes me feel a little bit more comfortable about saying this for transgender women. I still am not entirely sure about cisgender men. And again, clearly there's still debate, as there was discussion around this with some people who still think that the tissue concentrations are most important. I think people are aware there's a huge debate in this field whether plasma or tissue compartments mean the most. I'm definitely in the plasma camp. So I would rather a patient take a medication as best as they can and do it how they can do it, knowing that there's a chance they could, that they could end up seroconverting, but I'd like them to try. And if it means that they're going to take it less frequently, that's fine. So that would definitely be, a patient-to-patient conversation that I'd have of what people are able to do. And I think that's starting to come up more and more. I do know that there are still some providers who would never recommend anything less than 7 doses to transgender individuals. I think we know we operate in a harm reduction way. I feel very, I'm not going to say, so what if someone takes a tenofovir based oral regimen and seroconverts, but I am sort of going to say, so what, because they can still be on a 1-pill, regimen. So I'd rather them try and do it as best as they can and get them engaged in care. Maybe if I'm being blasé about this, I think our PrEP team knows, we haven't had any seroconversions. So maybe I'm relying a little too heavily on that, but, I think it's fine to do it that way.
Great. Any other thoughts or questions?
Thanks, Gabe. There was a comment about the issue of funding for injectable PrEP. And this is definitely a hot topic, and I think actually maybe better served for another time, as it's 8:59, but it is a big issue. And we know that cost is a concern, although it has been added to the Medi-Cal formularies. So that's good. It's just not on the PrEP app formulary yet. There's a workaround, but it's a difficult workaround. So I'll leave it at that, and we can talk about cabotegravir more in the future.
All right. Well, thank you, Dr. Jill Blumenthal, for presenting at our high rounds. And we hope to see you again in the near future. And thank you for attending. Next week. I
want to remind everyone, though, this is what I won for giving this presentation. Don't you want to get it?
So please let us know if you're interested in doing a talk. We're more than happy to accommodate. So thank you, everyone. Have a good weekend, and we'll see you next week.
Bye, everybody.